All right, so hopefully by now you learned that basically what Avery, McLeod, and McCarty did is they systematically destroyed each of the four macromolecules to determine if that macromolecule was the genetic material. So essentially, if you think protein's the genetic material, but you get rid of protein and transformation still occurs, then protein could not have been the genetic material, because without it, you still had the gene for smooth being transferred to the rough. Right? And same if you get rid of lipids, same if you get rid of carbohydrates, but as soon as they got rid of DNA, transformation no longer occurred. That meant that DNA was needed for transformation of the genetic material. DNA was the genetic material, the rest of them were not needed. And so basically, they did that by just lysing open the smooth bacteria after heat killing it. And obviously, what they would first need to do is just use the whole stuff that we haven't gotten rid of it and combine it with rough bacteria and see if they indeed got transformation, if you ended up growing some smooth bacteria, which would mean that some of the rough bacteria obtained the gene for smooth. And so you would need to do that prior to adding any of the chemicals that destroy certain macromolecules, because that's your positive control. You want to make sure that indeed your experiment is working, that transformation can occur if you have the cell lysate that is complete, has everything in it, right? Because if transformation can occur with the complete cell lysate, there's no point in destroying individual macromolecules because your experiment wasn't working in the first place. That's your positive control. But then they went forward and they took that cell lysate, they destroyed RNA, they destroyed protein, they actually did this with carbohydrates and lipids as well, and destroyed DNA. And in everything except for DNA, when they destroyed it, transformation still occurred. They still got smooth bacteria growing because the rough had been able to get the genetic material for smooth. However, when they destroyed DNA, that was not possible. And we know this because DNA is the genetic material. You get rid of DNA, there is no longer any gene for smooth that the rough can pick up. Transformation cannot occur. So this is how they showed that DNA was indeed the genetic material. However, like we said, people were still a little bit skeptical, right? Because there's a lot of other things that can go wrong. So Hershey and Chase really had the final nail in the coffin showing that DNA was the genetic material. And for this, they used bacteriophage. And bacteriophage was the perfect thing to use because what bacteriophage is a virus that infects bacteria. And so what viruses do is basically they insert genetic material into cells, live cells, in this case bacteria, but you have viruses that can infect your cells, a la the influenza virus, COVID-19, etc. They inject the genetic material into your cells and then use your cells to make more of the virus. So by injecting the genetic blueprint for the virus, your cell makes more of it. So bacteriophage kind of look like these little spaceships. They are made of protein with DNA in the inside, and they land on the bacteria and they basically inject the DNA, the genetic material inside. But this is perfect to use because a bacteriophage only has protein and DNA. So you basically only have two macromolecules that you have to differentiate which one is the genetic material, which one is being injected, which one is needed for the virus to put into the cell to make more of itself. All right, so bacteriophage being only protein and DNA kind of simplifies that. We can prove once and for all whether it's protein or DNA or maybe even both. So basically, here's what's happening. The bacteriophage lands on the bacteria. It injects the genetic material inside. Once the genetic material is inside the bacteria uses that genetic material to make more of the virus, which indeed ends up kind of killing the bacteria, but now there's a lot more virus there. And so they could try and determine what was being injected, what was the genetic material. Was it protein or was it DNA? And importantly, they use a label for sulfur and a label for phosphorus. Now, this is really important that they chose these two because this is going to differentiate between protein and phosphorus. So, if we think about it, we know we have that special amino acid that we had to memorize, cysteine. Cysteine has a sulfur side chain that is able to make our disulfide bridges. Thus, proteins, which are going to have cysteine as one of the amino acids, proteins have sulfur. But here's the key part. DNA does not. So if you label with radioactive sulfur, you're labeling protein and you're not labeling DNA. DNA, on the other hand, has the phosphate backbone, so DNA has a lot of phosphorus. but protein does not. So when you label with radioactive phosphorus, you're labeling the DNA and not labeling the protein. So the key choice of sulfur and phosphorus for this was because protein has sulfur, but DNA does not. DNA has phosphorus, but protein does not. So you were specifically able to label and therefore kind of visualize or record which one had the radioactivity, sulfur versus phosphorus. So in the first experiment, they radioactively labeled sulfur, right? And we know that means they're radioactively labeling the protein. So then they allowed the bacteriophage to grow or to infect the bacteria, and then they got rid of the coat that wasn't in there, and they basically were determining if there was radioactivity inside the bacteria. So if protein was the genetic material, then protein would have to be injected into this bacteria for the bacteria to be able to make more virus. But there was no radioactivity in the bacteria, even though more virus was made. Thus, protein could not have been the genetic material because the protein remained on the outside. It was washed away. The radioactivity of sulfur was never injected, therefore protein was never injected. On the other hand, when they radioactively labeled phosphorus and did the same thing, right? So in this case, the bacteriophage is only labeled phosphorus, right? It still has protein, it still has DNA, but the only label is the DNA this time. And so they gave it time for the bacteriophage to infect the bacteria, and then later on they saw that the bacteria did indeed have radioactive labeling, which meant the protein, or sorry, the DNA was inserted into the bacteriophage. The protein was removed, it was unlabeled anyway, so because radioactivity was only inserted when it was phosphorus that was labeled and not when it was sulfur that was labeled, they realized that the genetic material that was inserted was DNA and not protein. That was the final nail in the coffin, because if protein had been the genetic material or even was needed for that, this would not have been the case. The radioactively labeled sulfur would have had to have been injected into the bacteria. Protein would have to be injected, but because there was no radioactive sulfur injected, protein was not injected, protein is not the genetic material. On the other hand, phosphorus, radioactively labeled phosphorus, was injected. Radioactivity ended up in the bacteria when it was DNA being labeled. Therefore, DNA was deemed to be the genetic material. It was injected, protein was not. And even though protein was not injected, more bacteria were still made, which means protein could not have been, or sorry, more virus was made, which means the protein could not have been the genetic material. Because if it was, when you didn't have injection, when you didn't have radioactive sulfur inside, you wouldn't have the genetic material to do that. But because sulfur stayed outside, genetic material still got in, more virus made, protein could not have been the genetic material. It was DNA labeled with phosphorus. All right, so think about this. Would Hershey and Chase's experiment have worked if they had chosen carbon and oxygen? Why or why not? So think about why it was important that they chose sulfur and phosphorus, and the fact that sulfur labeled one but not the other, phosphorus one but not the other. So think about that and go ahead and answer that final question, and bring whatever questions you have on Wednesday to class, and I will be happy to answer. All right, good luck.